Okay, hi everyone. I am Eugenia and I am going to be the chairperson of our last block of oral presentation. Our first speaker is Viktor Hartung with quantitative analysis of citizen science data on Heteroptera, example of Allomorpha Alice distribution.
Okay, thank you, Chairwoman. Thank you, everyone. Peter Kment yesterday, you remember his very nice talk and very informative talk on Allomorpha genus, the taxonomy. He mentioned, he explicitly warned us against using citizen science when dealing with invasive species. I will try to make the opposite point today. But of course the difference will be, Peter was talking about tropical and subtropical countries. I will talk to you about Middle Europe. The situation is slightly different. So let's jump in. So the premise is that I was starting is that the citizen science platforms, and I was basically speaking about iNaturalist.org, they are very popular and assemble a huge amount of data, and they're transferred to GBIF and can get a DOI, and then you can cite them in a paper, and people do that. Like here you see, it's probably not visible from there, but it's like 2,500 different papers listed on the website of iNaturalist that use the data from iNaturalist. But most people use that for new faunistic records, like the new record from there, from here, which is a fine thing. But I just, at some point, I asked myself if there is something more to that. Maybe we could do something else. And invasive species, they are kind of very good, at least in Middle Europe, they're kind of very good objects for that, because if you would try to establish citizen science projects with seednets that Alex Ramsey is working at, I think that would be a nightmare. But these guys, they're mostly quite large, or at least in many cases large and conspicuous. They seek sometimes actively proximity to humans. And in case of Middle Europe, they are mostly quite easily to recognize, because we don't have this situation of Australia where another Allomorpha is coming to us. We just have the one. Yeah, so I tried to test on that something. And just before we jump in, I wanted to explain you the geography of Germany a little bit, because it's going to be important. So we're going to be concentrating on these two parts of Germany. These are the provinces of Germany, or so-called federal states. And they're very different in many cases. They have different governments as well, but these two, the state of Hesse is where I'm based at the museum in Frankfurt here, and the state of Baden-Württemberg. They will be especially important because these two are so to say the gateway of new invasive species coming to the country, because you see we have here the Rhine River, and here it is on the border, but here it is entering the country, and it is entering from a region that is very warm. So if you have some like southern European species or species from tropical, subtropical environments, here is where they mostly occur the first in the country. So that is going to be important later. Yeah, thanks to Salini and Lucia Adriana and Peter Kment, I don't have to tell you what Allomorpha halis is, because it has been described a lot. I just mentioned that in Germany it is known since 2012, and just as I said, the first place it appeared was in the very south here in Lake Constance. And then it started spreading northwards, and now we have Allomorpha almost everywhere, except for this very northern part of Germany, but I think it's just a matter of time. Well, just one thing that I wanted to test. There is this warm island hopping hypothesis on the spread mechanism of Allomorpha. So people think, and it's been quoted in German papers repeatedly, so people think that Allomorpha uses cities to spread. So cities are mostly a little warmer than the surrounding countryside, and this is good for a subtropical species like Allomorpha. So they first get from one city to another, and then if the countryside around is warm enough, they might spread outwards. But sometimes they just don't. Sometimes they stay in the city as well. So I heard that a lot, but I never saw a kind of reference or a study that would, like, pinpoint that. So I decided maybe I'd try to analyze the citizen science data, and let's see what they get. So this is what I did. iNaturalist allows you to export the data as an Excel file, and you can export up to 200,000 records, like huge amount of data. So I just I exported them, I selected them, uploaded them as a layer in GIS software, and then I just checked if the distribution points coincide with urban environments. Just a little details here, maybe some of you are interested. So this is the surface of the normal website of iNaturalist. Here we have the state of Hesse. Here is Frankfurt. And here are the records, the examples of them. And here you go to filter. Then you get this one here. And what I recommend to you, I'm sorry this is in German, but it's very easy to find in English or other languages as well. Here you have this research quality data, research grade. So it's better to use those. And then here on the right, download, you get to choose your columns that you need for your data. You can choose here the ID. You can choose the date, then it was recorded, and here, this is a place guess, this is the exact latitude and longitude, and the positional accuracy, which is also important, because sometimes they're quite accurate, sometimes not, but I come to that later. Yeah, and then you just get a GIS software. This is the layer where the urban environments are listed. So you can go to Carrenho land cover and download that for the whole of Europe. And I use slightly different source, but basically they are from this website. So this is a German offshot of that, so to say, and I just selected the layer for urban environments, and this is what you get. And then I uploaded another layer based on this exported file from iNaturalist. This you don't see much here, but when you zoom a little closer, you might see here little points. They are not very easy to see, but maybe you still can find them out. This one is slightly outside the urban environment here. But these are the very, very strict points. So I thought for a heliomorpha, a species that flies quite a lot and is quite active, it's better maybe to put a buffer around the point, like 100 meters circle, because they can spread maybe like 100 meters. So I just expanded the circles a little bit, and then I just counted how many times these circles, where the heliomorpha was found, are cutting the urban environments, and this is what I got. Almost all of the 200 records for the federal state of Hesse are the points that lie in urban environments. But you're probably already asking yourselves, if the points are located there, that means that the observers were there. So maybe this is not the bug that is restricted to urban environments, but the observers stay in there and just recording whatever they see. So what I did was checking it against some other species, because we do have more pentonomids, just the local ones, like Palomena prasina is probably the most widely recorded on iNaturalist. It has several thousands of records throughout the country. Or here, Raffigaster nebulosa. This is the species that is most likely to be confused with Heliomorpha halis. So I decided to check what you find. And as for Palomena prasina, you see it has similar number of finds, but only 63% of them are from urban environments. And if you fuse the points that are very close, like 200 meters away from each other, if you fuse these points together, then it is even 53%. So you see, in one case you have 95, and in another case you have 53. So it's definitely not the reason for Heliomorpha to be found in urban environments. Definitely not the reason for the observers staying in the cities is definitely not the reason for Heliomorpha to be found in urban environments, because they do find other species outside of cities. And if you compare it with Raffigaster nebulosa, it gets a little interesting, because it is similar to Heliomorpha halis. It also likes cities very much. And if you read the book, for instance, by Jürgen Deckert and Eckehard Wachmann, then they mentioned that Raffigaster is kind of fond of warm environments as well. So this is not a surprise when they shift to warmer environments. So I would say the warm island hopping hypothesis is kind of corroborated by the data from Hesse. But let's check, it's just Hesse. Let's check another federal state. Let's check Baden-Württemberg. And it's the same, basically. You have 95% almost for Heliomorpha halis to be in the city. You have less than 50% of Palomena prasina. You have Raffigaster, again, being very close to Heliomorpha. So I would say citizen science works with questions like that quite good. So I decided to ask, actually it was the conversation Leonidas Davranoglu and myself we had, that the idea came up with you know, this dynamics of invasive species. It's sometimes, I heard it described like that often. I don't have the source of this idea, but I thought of it myself like that. When the first invasive species arrive in some new environment, at first it's not noticed, then it kind of explodes and floods everything. It's like everywhere. And then after that, it might, you know, abate away a little, and it's not be so widely spread. So I just decided, if it functioned with this warm island hypothesis, then I would try to check with date occurrences of Haliomorpha and maybe some other species. What do they say? Oh, sorry, that was too much. Yeah. So now it's a table, and you have here 4 species. We have Haliomorpha here, we have Leptoglossus occidentalis, also very familiar to many of you, Oxycarenus lavatera, and Oxycarenida, and then this tingid Corytucha ciliata on Platanus. And you have here the dates they started to be occurring in Germany, the first finding. And unfortunately, you cannot find this out. It was actually the last 4 records have to be gray. So these last 4 are locals, and these 4 are invasive. And you can see in the beginning, it's like till 2017, it's not very much happening. Then 2018, there is much more records, and then the records began to grow rapidly, but not everywhere in the same way. Like if you compare all the locals, they just grow and basically do not stop growing in the number of records. But Leptoglossus occidentalis, Haliomorpha, Oxycarenus, Corytucha, they behave slightly differently, although I would suggest to omit Corytucha ciliata, because it's a tingid and it behaves differently, and you do find them in different locations, and they also do not have so many records for them. So I will try to present this in graphs. This is for the first not very easy to understand, but we'll try to pick it up, just piece by piece. So now we have all of the 8 species that you just saw together. This one is Corytucha ciliata, and you see it's just too few records, so let's omit that. It's still not very clear. But if you consider them just piece by piece, like first we take the native species of Pentatomida, this here, and you see they all have these dynamics. They either just go on growing, the number of observations just grows and grows, or at least it stays stable. Whereas if you take a look at the invasive species, this is Oxycarenus lavatera. It just falls off, and there are not so many records. This is Leptoglossus occidentalis, and this is Haliomorpha. And this is the only one that keeps on growing. But it happens when you have a look at the whole of Germany, and now I'm coming to a caveat that is necessary to remember when you work with iNatures, because the Germany in the idea of iNatures looks like that. It's a very square idea of Germany. That could be, sometimes it's okay, but sometimes it's a problem. So if you, when you work just state by state, province by province, it works pretty well, but here with this, it just covers west stretches of Poland, of Czech Republic, Austria, Switzerland, whatever. So, but if you have a look at this graph, if you look at the records for Haliomorpha halis for Baden-Württemberg alone, then you also see it also does fall off a little bit. So I would say basically the pattern for the invasive species is they've been growing 2017 until 2020, and then they go back, whereas the native species, they just keep on growing. The observations just grow and grow. Why? Because 2018 till 2020 were the 3 drought years, what they're called in Germany. It's been massive droughts. It was very dry and very hot. It's not very visible here, I'm afraid, but this is like a kind of normal situation. So this mostly green, and this one is only slightly yellow. This is 2018. And yeah, unfortunately, the colors just do not really come across here, but you have to believe me, it's much more yellow in this case. And if you, sorry about that, and if you compare, oh yeah, you're right. And if you compare the precipitation for the same periods, you also see this pattern. So the precipitation here drops off massively. Whereas 2021, after these 3 drought years, it goes quite well back to normal. So this one is already very similar to that, but this 2018, 19, 20, it was very dry and very hot. So my explanation would be for the invasive species is that they come from warmer environments mostly, and they kind of better feel themselves when it's warmer, and when it's getting colder again, then the records and the occurrence of the bugs goes back. But of course, there is still more work needed to do. So we see that there is some abating of these records. It might have something to do with the climate, but I still have to run more tests. I've been working on that like in the last days again as well. So it's still the work ongoing. But I think there is something to this pattern. And well, the more tests is needed, more research is needed every time, of course. And well, actually, I'm almost done with the talk. For now, I just would like to present you some caveats, because I think I made my point that the citizen science records, including iNaturalist, which has the best system to export the data, to my mind, definitely have a potential. But you do have to bear in mind these country borders. I do hope the programmers of iNaturalist maybe fix this problem, because I don't think it's a very hard thing to fix, because the province borders, they are right, just the country borders that are not. Taxonomic accuracy. Because it's better maybe I did check the identifications of the species when I was working with that, but sometimes you might have, well, maybe too many records, so there might be some procedure needed to check for taxonomic accuracy, but you have to remember iNaturalist actually has this kind of crowd, swarm intelligence way of doing this, because they need 3 confirmations for the ID before it's like research grade quality. So it might not be that big an issue, but still you have to bear that in mind. Positional accuracy, because sometimes people record the position when they make a photo of something, then it's very exact, sometimes not, and in some cases the positional accuracy is written, in some cases not. So you have to deal with that as well. And also there are some double records. So some people just, they see 5 animals and they make 5 photos and they upload them all at once. So it's, I would consider it 1 record, but they, in iNaturalist you have like 5. So there have 5 lines in an Excel file, which also has to be controlled for. But still, I think I've made my point, and I believe this is quite a very good source of the material and data, and I think it has some potential to not only for faunistic studies, but also for studies of biology of the bugs. And yes, thank you very much for your attention, and thanks to all who contributed on iNaturalist talk. Thank you.
Okay, is there any question? Yes.
Congratulations. Have you considered the increasing use, when interpreting the temporal dynamics, have you considered, or how could be considered the increasing use of digital photograph by people, by citizens, and when and also together with this, the increasing knowledge by citizens of the increasing taxonomic knowledge. I mean, for example, if people learn to recognize the invasive species, they want to photograph it, for example.
Well, as for the increasing knowledge of the invasive species to influence in that, I think that is that would argue against, because if they're still interested, and it was growing, and they're still interested in the native species, because this graph, it still goes up, but this one falls off. So I think that's not the interest. I think the interest is there. They just see less. As for the amount of the digital technique, I think that's what happened here, because it's where it starts to grow rapidly. But of course, it's hard to separate in this case. It definitely doesn't have to do with bugs becoming so numerous. I think this increase here, 2017, I think it's definitely because of the availability of the digital technique and maybe smartphones that are capable of doing research grade photos. I think it's like that, but this is just my guess for now. I cannot really pinpoint that.
The last one?
Viktor, I just want to make a comment.
Sure.
I was just going to make a comment. You're using this to monitor for particular species. We've had several instances in Florida just recently where, through iNaturalist, we've found bugs that have been introduced that were off our radar, and we've sent people to confirm that that bug is there and that it is the bug we think it is. So we've had some invasive species, or at least immigrant species, show up that we weren't aware of, and this alerted us to where those bugs were. One of them's Brachyplatus sebaneus, which has the potential to be a bad pest and a bad invasive species. So we get to it a lot quicker. So there's some real advantages to this.
Yeah. Sure.
Okay, sorry, but we have no more time for question. Okay. Our next speaker is Renan Carrenho with Conspectus of Dellapé of the Americas.
Hello, everyone. So let's talk about reporter chromids. This is a classic tray of reporter chromid. Maybe some of you just find like just boring, dirt covered, but actually under microscope they're quite interesting. Reporter chromids is within the superfamily Ligioidea. It has more than 2,000 species worldwide, and the tribe Lithiine that I'm working on right now is the most is one of the most diverse tribes of this family. It has 41 genera and 190 species worldwide, and in a new world, like in Americas, what's my work is focused on, has 54 species in 13 genera. This tribe is considered monophyletic based on the linear position of Dicobotia in the 5th abdominal sternite, extremely modification of the stern reservoir, development of iridescent areas in the head, and the presence of Dicobotium at each lateral pronotal corner. Here we can see more closely the iridescent areas. Let's call them patches here. They're like two kinds of patches. Two patches here, and just one spot. This is quite interesting for this group. And when you see the distribution of these characters in the world, we see that in the old world we see just two patches, and a new world we see one patches and two patches. For this, for the Americas, there are two main works that there's a lot of new distributions and revisionary studies, like Baranovsky and Slater in 2005. They studied these bugs in West Indies, and Pablo Dellapé and contributors. Sorry, I'm really dry. They studied these bugs in Argentina. These two works will reflect on the distribution of the current knowledge of these species. We have in Brazil a project that we collect in Caatinga. This is a biome that is like a dry forest, and we did 8 expeditions there and collect all order Hemiptera. And in addition to that, I've been visiting some collections. For instance, IMPA in the Amazon state, in Curitiba, and Smithsonian. With this, we found new records for 14 species, country-wise, plus several state new records. Pay attention to the West Indies and Argentina. They are always there because of the only many works they've done to this tribe, with this tribe. Bubastis castaneus occurs in all these countries, and we found in Belize. Cistalia, we added Brazil. Crifula fasciata, Honduras. Gonatoides typicus, Brazil. You see, for this example here, they're only the north area of South America and Central America, and sometimes they occur also south, like this one, like Argentina. Lipostemata humularis, Lipostemata major in Ecuador. We found this new record. Neopetitius, Freshneri, Ecuador too. Paragonatus costaricensis. We have a lot of new country records. Paragonatus divergens. Also, Belize, Brazil. It's always where they collected the species. Another one, Brazil again, Brazil, Bolivia. And about the biology of this species, these specimens, this species of these tribes, this tribe, it's hard to predict. Once they are, when you see the labels and when we go to the field, we always collect by light trap and malaise trap. So we just know they fly well. But we have a case of the genus Lipostemata. Here were found on aquatic plants, and in this project we were able to collect them in water and also in cistern. So this could be semi-aquatic, maybe. Reaper chromid. This genus was once recognized as a division by Berg because they're completely different than the other lithiines. The lack of dicobotium on each side of pronotum margin, here, like right here, and O'Donnell treated as these characteristics as plesiomorphous. So are these plesiomorphous or specializations for semi-aquatic environment? So also we have some questions related to that. So do Americans genera and the current generic classification represent monophyletic groups? Are the genera with 1 or 2 iridescent spots natural groups? What are the relationships between the American genera? In order to access this these questions, we are starting doing a preliminary analysis with a morphological approach. We collected 54 terminals with 52 characters. So in group, we have 32 species from neotropical and Arctic regions, and 12 species from Afrotropical, Australian, Indo-Malaysian, and Palearctic region. We had all the New World genera, but Isurs. And outgroup was 4 tribes, other than the Lithians. We did that just to try to just see if they have this different pattern between Old World and New World, and the 1 patch and 2 patches. So this tree shows us that here all this clade has 2 patches, and this clade here has one patch. So that's we was expecting this once the 1 patch would be plesiomorphic. Oh, that's not good. So this another example of the they need revision, because they're really similar, and they are like Crifula, Paragonatus, and gonatoids are really an interesting group, but they are really similar between them. Lipostemata, O'Donnell said that would be a plesiomorphic characteristics, and here we see that's the end of the tree, so could be some kind of specializations. The characters could be for specializations. So we always have more to do. So, like I said, we have to revision at generic level, find more morphological data, and with the field trips we collect also in alcohol, so we can access molecular data, and hopefully soon we have more things to share. And thank you all.
We have time for questions. Someone?
So iridescent patches are in other groups in Heteroptera, but what do you think the function is for these guys? Any speculation?
Yeah, actually this patches has 2 main structures, different structures. They can be like with stripes, and they are really rainbow when you see under the microscope under the light. And the other characteristic, the other kind of patches could be also really blurry with like little dots. So they also had different, I don't know if they're, why they have these specific differences. Actually, I don't know about this hypothesis again. Yeah, good question.
All right, so I saw you had Cryphula abortiva was sister to Paragonitis costaricensis, and they collectively were sister to your remaining Cryphula. Did you have Paragonitis divergens in that tree?
Yes, it is. Right here.
And where is?
Cystalia, and Paragonitis costaricensis right here with Cryphula.
Okay, follow-up question. What's going on there? Because I'm familiar with both, because we have Costa Ricensis and, well, just in the Keys, and I've caught loads of divergens over the years, and I've seen them side by side. I never would have thought of that. Do you have an opinion on that?
Yeah, I mean, definitely we need more characters to define like more precise this. But yeah, every time I see Paragonatus costa-ricensis, like if you see the pronotum and the general characteristics, you see like, is this a Crifula or Paragonatus?
And those are the only two species in Paragonatus, correct?
Yeah,
okay.
only these two. Yeah, in Crifula, there are a lot of species. Yeah.
Okay, I approve of this answer.
Someone else? No? Thank you very much.
Thank you.
Our next speaker is Zeman with hidden life in the interstices on biology and taxonomy of Dipsocoridae occurring in Central Europe.
Good afternoon. So my name is Šimon Zeman. I'm currently starting my master thesis at Charles University in Prague, Czech Republic. And today I would like to tell you something about a tiny little ongoing side project we have with Viktor Hartung and Kment about dipsocorids in Central Europe. So as you may know, dipsocorids are similar to other families from the info, the Dipsocoromorpha, rather small, brown, a bit beet-like in appearance, as you can see here. It doesn't seem to work. Yeah, it does. It's just a bit slow. Okay, so in Central Europe, 5 species were thought to occur. 2 species of Pachycoleus, 1 species of Alpagut, which, however, is just a single record from Hungary, and 2 species of Cryptostemma. Cryptostemma carpaticum, being confined mostly to the Carpathian area, which is in Poland and Slovakia, and Cryptostemma alienum, which was thought to be more widespread. However, after a careful examination of specimens from my collection and from collection of National Museum in Prague, we found out that there is another species that was confused with Cryptostemma alienum, and probably in a lot of cases not identified correctly. And that is Cryptostemma rehmanni, which was formerly thought to occur only in the Mediterranean area. So how to tell the Cryptostemma apart? This, what you see here, is the dorsal side of abdomen of a male. You can see that there is a lot of appendages arising not only from the genitally capsule, but also from the tergites. Those are the so-called paratergites, and this bizarre structure probably serves as some kind of a saddle for the female during mating. This, I think, was described for Ceratocombus, which is from the family Ceratocombidae, but in Cryptostemma it has not been described yet. Anyways, these structures are very handy when it comes to identifying Cryptostemma because they can be reliably identified only based on the male genitalia. From Cryptostemma carpaticum, Cryptostemma rehmanni can be distinguished based on this paratergite 8. As you can see, the difference is quite noticeable. And more importantly, Cryptostemma rehmanni can be distinguished from Cryptostemma alienum based on the shape of the 6th abdominal paratergite. Here's a picture what it looks like, and you can see that, again, the difference is quite noticeable. When Josifov described the species, Cryptostemma rehmanni, he was not sure whether it's a valid species, but at least from what we've seen so far, we haven't found any intermediate specimens where we wouldn't be sure whether it is Cryptostemma rehmanni or Cryptostemma alienum. This is a map of places where we have found Cryptostemma rehmanni already. As you can see, it includes Germany, Austria, and also several places in Czech Republic and Poland. To be honest, there was a find that was on the very border with Poland. It was on the stream that was making a border between Poland and Czech Republic. But we found it both on Polish side and Czech side. So that's a record for 2 states. What is interesting, however, is that Cryptostemma rehmanni seems to be more widespread than Cryptostemma alienum. We still have a lot of material to go through, our own material, and especially a lot of material from the collection of Professor Pavel Štis. But from what we've seen so far, it seems that Cryptostemma rehmanni is either as common or even more common than Cryptostemma alienum. So, now about the biology of the 2 main dipsocorid genera in Central Europe, Pachycoleus and Cryptostemma. Cryptostemma inhabits gravel riverbanks. It's quite a specific habitat. It has to be sunny, because we have never found Cryptostemma in any gravel banks that would be shaded by trees or be in a wood, etc. And it also has to have a proper type of substrate. You can see it includes both larger pebbles, smaller pebbles, and a sandy soil. And between those pebbles, a tiny little spaces occur, which are called interstices, and that's where Cryptostemma live. And that's why they crawl and look for their prey. The soil can be a bit muddy, but not too much, because otherwise the clay particles can damage the waterproof surface of the bug quite easily. And this kind of habitat is in Czech Republic confined mostly to higher altitudes. And so is Cryptostemma. But we don't think that Cryptostemma is a mountain species, but it's just because that in lowlands, this kind of habitat, naturally flowing rivers that are not straightened, that didn't have their dynamics altered, just don't occur anymore in lowlands. So they are mostly in the mountains, and so are both Cryptostemma rehmanni and alienum. Pachycoleus, on the other hand, lives in some wetland habitats like bogs, spring meadows, fens, especially when there is a sphagnum moss present. And between the sphagnum leaves, again, there are the interstitial spaces where Pachycoleus lives. Both of these habitats have something in common, and that is that they are often flooded. And its inhabitants, they have to cope with that somehow, and they truly can. Both Pachycoleus and Cryptostemma can survive underwater for several weeks, in case of Cryptostemma, even for 2 months. And they can do so because of this silverish air layer that forms around their hemelytra and probably serves them as a kind of a plastron. So it's a storage of air that they can breathe during the time they are submerged. The formation of this layer is mediated by the surface of hemelytra, which on both upper and on lower side is covered with a tiny little microtrichia, which trap the air and make it such a nice air bubble. What is interesting is that Cryptostemma and Pachycoleus seems to have different feeding preferences. Cryptostemma seems to be more of a scavenger, meaning when it encounters a live prey, it usually ignores it. It goes around most of the time, touching the surface with its rostrum, and when it finds a dead insect, it starts feeding on it. Here you can see it feeding on a dead mirit. Yeah, this is Cryptostemma feeding on a dead grasshopper. Of course, it didn't hunt it itself because that would make it an apex predator, which is surely not. And even the rostrum itself is very, very strange. As you can see, there is a cavity in here that is full of various olfactory sensilla, which probably enable Cryptostemma to look for the dead insects to feed on. But the most striking thing is these leaf-like sensilla. I've never seen that in my life before, and unfortunately, I do have no idea what purpose does it serve. We thought it might be something olfactory, but in closer magnification, there were no pores, so we just don't know. On the other hand, Pachycoleus seems to be more of a predator, because when it encounters a live prey, as you can see here, it's a tiny little collembolan. So when it touches it with its four tarsi, it immediately snatches it and devours it. And if you have a look at the tip of the rostrum in Pachycoleus, it looks completely different than in Cryptostemma. There is no cavity, and in the lateral aspect, there are also none of these striking leaf-like sensilla. As a matter of fact, its rostrum looks more like the rostrum of Ceratocombus, which is from a different family, but is also well known to be a predator. Yeah, and here you can see the comparison between labium of Pachycoleus and Cryptostemma, and you see that the difference is quite striking. Now, in order to confirm these hypotheses about feeding preferences, we have to keep the dipsocorids at home or at lab. For us, it worked quite well to keep them in, at least Cryptostemma, in tiny transparent boxes with some soil that are wetted every 3 to 4 days. And you can feed them basically with any dead insect. It even feeds on dead acari. It just doesn't like dead earthworms. For Pachycoleus, it's actually even easier because they can be kept in such small glass vials, which are filled with water. And there is a cotton plug, and above it is a tiny space where the Pachycoleus lives. You don't have to moisture that even because the water there provides enough moisture. And again, you can feed the Pachycoleus with either dead insects or with live ones. As I mentioned, this is an ongoing project, so we still have a lot of work to do. First, we would like to find out whether Cryptostemma rehmanni is actually a valid species. For this, we want to obtain barcodes of both Cryptostemma alienum and Cryptostemma rehmanni. We've already done that for Cryptostemma rehmanni, but the problem is that in the past 2 years, we haven't been able to obtain a single specimen of fresh Cryptostemma alienum. But as I said, we still have a lot of material that we've collected and haven't gone through. We hope that at least some Cryptostemma alienum is going to be in there. Does Cryptostemma rehmanni differ from Cryptostemma alienum in its habitat preferences? We don't know, but we surely want to have a look at that. And we definitely want to test and properly verify the hypothesis that the Pachycoleus and Cryptostemma differ in their feeding preferences. And in the course of doing that, we hope to enlighten what is the function of the crazy labial setae of Cryptostemma. And if it's possible, we would like to have a look at many other things, like mating behavior, which we expect to be really crazy, the ability to survive underwater, how they even overwinter, and internal parasites like the trypanosomatids, whether there are some. But first things first, we have to revise the distribution of Cryptostemma in Central Europe. So this is the place where I would ask you for help. If you have any material of Cryptostemma from Central Europe or from Europe in general, please be sure to check it out using the Josefovsky, or you can send it to me for identification. And please let me know whether it is really Cryptostemma alienum or Cryptostemma rehmanni. Furthermore, if you know about any locality where Cryptostemma occurs and you can go there, collect it, bring it to lab and observe it, please, I'll be very grateful for any observations that would be about feeding behavior, mating behavior, overwintering, etc., etc. And if you don't know about any locality with Cryptostemmas, I recommend you that you look for the habitats that I mentioned. River gravel banks, sunny, with a specific type of substrate. You lay down in there, basically any time from April to October, and I think you have quite a high chance of finding the Cryptostemmas in there. So with that being said, Cryptostemma rehmanni occurs in Central Europe, and seems to be at least as common as Cryptostemma alienum. But Pachycoleus and Cryptostemma both inhabit different habitats, but both of them have to survive underwater for several weeks or even months. And probably they're enabled to do so by the plastic structures that envelops the hemelytra. And hypothesis, while Cryptostemma seems to be more of a scavenger, Pachycoleus is truly a predator of small invertebrates. And that would be it. Thank you for your attention, and if there are any questions, I'll be happy to answer them.
Questions?
Thanks for this wonderful presentation. Do you have any plans on also looking at alpagut at some point?
Once again, please.
Do you have any plans of also looking at alpagut at some point, maybe?
Well,
because it would
if
be
we
just
find it,
wonderful to, do the same stuff with those.
Well, as far as I know, that there's only this one record of alpagut in Central Europe, and I do not have, I haven't never seen that in my life before. So if we ever stumble upon it, we'll sure do something with it. But until then, yeah.
Someone else? No? Okay, thank you so much. Okay, our next speaker is Renan with Heteroptera diversity of the Fernando de Noronha archipelago, Brazil.
Hello again. Now we're going to talk more a broader group and the Heteroptera diversity in this archipelago. This archipelago was discovered in the early 16th century. It is located in the equatorial South Atlantic region, northeast Brazil. It's approximately 106 kilometers of the nearest coastline in the northeast of Brazil, and it is part of state Pernambuco. And also, it's a preservation area. I've never been there, but I know it's for rich people, so it must be a really interesting area. It's in this. Located in that area. It's kind of far from the coast, and it is entirely volcanic, never connected to the mainland. It is a small archipelago, but there's a main island. It has 16.9 kilometers squared, and plus 20 smaller islands. The biggest one is Hata Island, like 1 kilometer square. The habitats there is basically Restinga, which is like the coastal habits with the small bushes. In general, aspects of this archipelago is in 1737 was built fortifications and became a penal colony for almost 200 years. So we had like a larger occupation since then. Also in World War II, that impact the island with introduced animals, plants, and also use the insecticides. That also they removed a large part of the native flora and they introduced hundreds of exotic species. But also it still has, I think, approximately 32% of endemic plant species. What about entomological fauna? First records there were for Kirby in 1890, and he found 57 species of Coleoptera. Waterhouse in 1890 found 21 species of Coleoptera, and Hidley the same year 5 species, the Zanura and Collembola. In 2020, Rafael collaborators estimated that the island has 453 species, including other groups like Collembola. So finally, Heteroptera. In 1957, Carvalho Becker described the first Heteroptera for their archipelago, which is a myriad. They also reported 2 myriads and one Hinacloa was just one specimen. He couldn't precise the species. And then Alvarenga in 1962 added a new record: 1 pentatomiid, 1 rupalid, 3 riperchromids, and 1 validid. So this is the species described by Carvalho there, and this is the possible species that he collect there, Hinacloa. He also mentioned that he possibly be this species, but just one specimen, he couldn't tell. So in 2019 and 2020, Rafael and collaborators, they went to the island and collect all orders of insects. They used active collection methods such as sweep nets and just trying to catch them by hand. And here I think he's just using some kind of knife to get some under the soil. Also they use passive collection methods like malaise traps here in a more dense forest, and here in the mangrove close to the coast. Also Pennsylvania light trap and conventional light trap. So after that, they published this results of this collection, and with regarding to Heteroptera, they collected more than 20 species of ctenids, 20 species of ligades, and more 20 specimens of gingids. This is the article, and the new records for, and we got this all the material that he sent to us, and we got in green there are the new records that we recorded. So Anthocordy, we have 3 species, Beritid, we have 1. So Ctenidae could also identify as Delacielos, Vidas. Ligade was Nisias, 2 species, Myriad, Cytocarpus, Femoralis, two species of Stenodemini, and another one of Nascorini. One nabid, Pentatomid was Endecini, and Hopalid was a Hermostes gravidator. And Theochrorid was a Galgufa, and Tingid, we got also 3 species. We'll see them now. Like these are the Anthochorids. So I'd like help to identify them. These are the Mirids, the Cytocarpus femoralis, and these 2 Stendonomini. So they also collected, again, the Hinacloa, probably would be this one, and also species of Nabidae. These are the 3 Tingids, Varica, Corituchka, and Gargaphia. Bericidi was Gampcoris nexus. These 2 Nisias, they're really, I find like really hard to identify them. 2 species. And there's 3 Reperochromids. The Jadera that also described for the archipelago. Galgufa, Tyrocoide, and the Sidinid. The Edesini, there was this one. Tianta for the Pentatomid. And what we got from this last field trip, the survey, that was basically that we increased the number of species for the island. So we had some surveys there, but as you know, it's never enough. So Jadera, the Hopalidis Jadera, Cortunix, Hufulux is endemic to the archipelago, and Jadera Cortunix is found from Texas to Argentina. Garganus insularis was described also for the island. Quiotinus pallidus is found all around the world, from Brazil to Yemen, including Africa and Europe. The Mirid, Hyganochlora clavicornis, is found between Mexico and Argentina, including the West Indies. So what are the questions then? Are there more species endemic to the archipelago besides Jadera? And when and how they colonize the islands? How widespread are the species found in there? And was any species man introduced to the island? These are hard questions to answer, like mainly when we are talking about arthropods. We have this example of the Amphisbena, that it is endemic from the archipelago, but like as vertebrates, they have more data available, so they can explore more easily. In our work with genomics, and then they had like discovered that this hypothesis of the colonization first was through West Indies and other Paleo Islands in the Caribbean, or Amazonic orange, and was just swimming or fluctuating with something until the archipelago. So could any true bug have colonized the island through similar process? Even though insects can fly, colonization via Western US product could be a possibility. Also, as I think another hypothesis would be, of course, that they just follow the host plants, but we need to get more information of their introduced plant species there related to the Heteroptera found there that's with the host plants. And that's it. Thank you.
Questions? No? Okay. There.
If I have any other things from there, would you like records?
Sorry, I didn't hear.
If I have material on loan that's possibly from that location and it's not included in your list, would you like to know about it?
Yeah, of course.
All right, because I'm pretty sure I have a series of a different Gargaphia species.
Okay, great. Thank you.
Other questions? No? Okay, thank you. And our last speaker is Randall Schuh with Fieldwork and Phylogenetics, Advancing Miridae Biology and Classification.
Good afternoon. So my presentation today will tell a retrospective story about how we have increased our knowledge of the Miridae subfamily Phylini and the approaches that have been important for doing so. And it will include some personal details about me and the people that have worked with me. By way of background, I grew up in the western United States in the town of Klamath Falls, Oregon. My father was an entomologist from whom I learned a lot about insects as a child. And I also became acquainted with many entomologists who visited our home during my childhood. One of those entomologists was Jack Lattin, who you see in the lower left here, who was then a young professor at Oregon State University. I first met him in 1958 when he brought a class on a field trip to Klamath Falls, and you'll see some of the people that were on that field trip, including Jack, my father, and me, at a much younger age. He later employed me at the Oregon State University Entomological Museum during my 4 undergraduate years there. He also gave me the opportunity to serve as a summer intern in 1964 with funding from the U.S. National Science Foundation to study the Saldidae, a group that he had become interested in at that point. That summer was the beginning of my interest in academic entomology as a potential career path. In 1967, I was accepted to work on a PhD with James Slater at the University of Connecticut. I spent the summer months dissecting Blissidea genitalia, the group of Slater's interest, and in October we departed for 8 months of field work in South Africa, where I gained experience in collecting methods, host plant documentation, South African geography, and the field biology of Heteroptera. As a dissertation subject, I chose to work on the Miridae because of suggestions that had been made to me by Jack Lattin. I'd worked on the Saldidae as a master's project, a more tractable group for a beginner. The Miridae group, as you can see in this slide, of about 11,400 or more species with other attributes as outlined here. About 3,000 of those species belong to the subfamily Phylini, which is the subject of the focus of the rest of this presentation, although I will mention some other mirids. My dissertation work focused on the fauna of South Africa. At the time, only about 40 species of Miridae were recorded from the region as recorded in a South African Animal Life fascicle that was co-authored by José Carvalho. 14 of those species belong to the subfamilies Orthotylini and Phylini, which is what we're going to hear more about. My published work treated 103 species in these two subfamilies, 81 of which were described as new, a strong signal of the need for more fieldwork and taxonomic study of the fauna of South Africa. I attempted to emphasize the phenomenon of ant mimicry, but I soon realized that many taxonomic problems needed to be resolved before any real progress could be made in addressing mimicry in a substantive way. The Carvalho catalog was instrumental in facilitating my research. It was published in the 1950s, beginning in 52 and ending in 1958. It facilitated my research, but nonetheless, the classification presented in it had some serious limitation in the subfamilies of my particular interest, as pointed out on this slide. The Phylini weren't monophyletic, the Orthotylini weren't monophyletic. Many genera were commingled across the two subfamilies, and recognition of defining characters for monophyletic subfamilies and tribes, including genitalic features, were lacking. My first attempt at phylogenetic analysis treated the restructured and putatively monophyletic Orthotylini and Phylini as sister groups. These conclusions involved the inclusion of characters from the male and female genitalia, attributes which had been documented by Wagner, Slater, Kelton, Carvalho, and others, but were not used by Carvalho in developing his classification. In February of 1974, I was invited for a job interview at the American Museum of Natural History in New York City. I think the second time I'd ever been in New York City, a place about which I knew very little. I had the good fortune to be offered a job and began work in September of that year. I was one of 5 entomologists on the staff of 40 curators. At the time, the American Museum, usually known as the AMNH, was a place of great intellectual ferment in the fields of phylogenetic analysis and biogeography. And for me, it was a very exciting time, and like going to graduate school all over again, but a lot better in many ways. At the American Museum of Natural History is the second largest natural history museum in the United States, following only behind the Smithsonian Institution in Washington, D.C. Entomology has long played an important role in scientific studies at the institution, which has arthropod collections in excess of 20 million specimens, and today those specimens include about 1 million specimens of Heteroptera. When I arrived in New York, the AMNH Miridae collection consisted of about 65 drawers of specimens. 2/3 had never been sorted to any level except Miridae. And most of the ones that were sorted and identified came from the collections of Meyer Dürer, the Swiss entomologist from the middle of the 19th century. Today, the AMNH Miridae collections exceed 1,000 drawers, with about 75% of all the material sorted at least to genus. Confronted with the very limited collections and the need to develop a viable research program, I was forced to ask, where should we do more collecting? To provide some additional perspective on the Miridae, here we see species accumulation curves for the major zoogeographic regions. From the late 1960s to the early 1980s, the Nearctic, Neotropical, Ethiopian, and Oriental regions all received a significant increase in known species level diversity, important papers that contributed to that knowledge being indicated on the slide. Nonetheless, these areas all lag behind the Palearctic region and the long history of studying its rich fauna. The possibly most notable on this slide is the poverty of knowledge for Australia, or at least apparent poverty of knowledge, a subject of which I will have more to say in a few moments. The Miridae show substantial host specificity, greater than any other group of Heteroptera, certainly. This graph plots the numbers of Miridae species against numbers of known hosts. Most species occur on a single single plant species, as shown in the vertical bar on the left of the graph. Miridae species with increasing numbers of hosts trail off to the right until we get up to 12 or 15 possible hosts. These data suggest that when collecting Miridae, we should document the identities of the plants on which they were captured. It's certainly the simplest way to know where to look in the future if you wish to recollect a given species. In the spring of 1978, Tom Henry, our president, who unfortunately could not be with us, invited me to go with him on a trip to Mexico that was to be led by Joe Schaffner from Texas A&M University. We spent 2 weeks in Mexico and West Texas, where we collected many Miridae. I collected for another month in the southwestern states and the Rocky Mountain states, and when I returned home, I had about 5,000 specimens of mounted Miridae, my first major attempt to collect the group in North America. In 1968, the godfather of North American Miridae, Harry H. Knight, published a large paper whose cover is shown on the left of this slide. It was greeted with great fanfare by Jack Lattin and others because the Western North American fauna remained poorly known, in contrast to that of the rather distinct Eastern fauna, which was much better known. Although Knight's 1968 paper treated in a nearly comprehensive way the Miridae fauna of Western North America, I found it difficult to use for identifying many taxa I had collected on the trip with Tom Henry because it lacked habitus and genitalic illustrations for most taxa, features that I had learned were vital during my dissertation research on the South African fauna. In the spring of 1979, I returned to Oregon State University at the invitation of Jack Lattin to spend a week working with Jack's students who were studying the Miridae, of which there were several. Among them were Michael Schwartz, Gary Stondahl, and Jerry Cassis, all of whom I met for the first time. At that time, I also made the decision to begin serious study of the Miridae of Western North America. And this decision was influenced by a simple question put to me at the time by Jack Lattin, which you see quoted on the lower right. He said to me, "Toby, how are we going to identify all these Miridae from Western North America?" I had now realized that the situation in Western North America was little different than the one I'd encountered in South Africa as a graduate student, and then the answer to Lattin's question had several parts. Among them are the following. We needed to do much more field work. We needed comprehensive generic revisions. We needed the publication of habitus illustrations. We needed the consistent illustration of male genitalia was done by Edward Wagner in all of his post-World War II papers. And we needed to document host associations in a more consistent and precise way. So what can be the impact of fieldwork then? I went on to organize and participate in 10 more field expeditions in the western United States and Mexico. And here you see one shot from the field in southern Arizona. This map shows the localities we visited and from which specimens used in subsequent revisions were collected. My thanks to Michael Schwartz for making the maps and for participating in many of those expeditions. Our fieldwork contributed to numerous publications that greatly improved knowledge of Western North American feline Miridae, including the description of more than 135 new species. Including 3 of those shown here for the genus Tuxedo, the previously described species of which had been placed in several genera by their authors, Van de Zee and Knight. In early 1993, my wife Brenda and I were eating dinner in our New York apartment with visiting Australian colleague Jerry Cassis. Jerry said unexpectedly, "How would you like to do some field work in Australia? I think I can get some funds from my employer to cover part of your expenses." Well, I said yes, and eventually we took 7 month-long field trips in Australia. Jerry and I collaborated in our record keeping to begin documenting on a large scale and in a very consistent way, the Miridae fauna of the continent, including hundreds of host plants from the rich endemic flora. And one of those host plant groups is the Proteaceae, and Jerry's standing next to one of the species of Grevillea. In 2003, Jerry and I received funding from the U.S. National Science Foundation to study the Miridae subfamilies Orthotylini and Phyllini on a world basis. Our request for funds was based heavily on our fieldwork that we'd already done in Australia, localities for which are seen in this map. Jerry continues to lead additional major expeditions in Australia, and every trip that he takes results in the discovery of more previously unknown species of Miridae. Although much work remains to be done in Australia, much progress has been made, as described in the red. In the black is a partial list of publications on the Phyllini by me and several of my colleagues. These representing an 800% increase in the known Phyllini for the continent, but with many well-documented taxa still awaiting study and description. An equivalent list exists for the Orthotylini, the result of work by Jerry and his colleagues. Our National Science Foundation grant also funded 3 field trips to South Africa in September and October, the optimal time for collecting Miridae in the southwestern Cape floristic element with its Mediterranean climate and remarkably rich flora, but one which I had only visited during their summertime when I was a student during the 8 months I'd spent in South Africa much earlier in my career. We now know that the Phyllini fauna of the Western Cape is extremely diverse, although the Orthotylini is somewhat less so. This map plots the localities where we collected in 2003, 4, and 7. I will describe 2 studies that exemplify the impact of our 3 trips to South Africa. First, using specimens we had collected, NSF summer intern Grace Wu and I revised the genus Eminoculus. You see one of the species in the lower right. All 7 known species of which feed on the endemic Mesembryanthemoideae, sometimes called ice plants. We described five of those species as new as a result of our field work. DNA sequencing work done by Katrina Menard showed that Eminoculus is actually a member of the myrmecomorphic tribe Halodipini. I had described Eminoculus as a new genus in my dissertation, but placed it in the omnibus tribe Phyllini. At the time, the only known host, and the only known specimens, were from ornamental plantings in Pretoria. AMNH employee Ruth Salas and I revised the sister genera Pseudocinaris and Parapseudocinaris, all 14 species of which feed on members of the South African endemic group Crotalaria in the family Fabaceae. Whereas I'd placed these genera in the omnibus tribe Phyllini in my dissertation, Katrina's DNA sequencing work placed them in a reconstituted tribe Simiini, a long unused name originally associated with the Orthotylini. I'd previously seen only a few specimens representing 5 species, one with host data. We examined more than 8,000 specimens from our own NSF-funded field trips, nearly all of which had host data, and we described 9 new species as a consequence. Now let's take a look at the power of phylogenetics. In the Carvalho classification, the 4 large subfamilies of Miridae, of which we've heard some in previous talks, including the Phyllini. Each contained one tribe recognized by its ant-like appearance. Each of them also contained a nominate tribe that lacked defining characters. That would include Mirini, Baryocarini, Orthotylini, and each of them lacked defining characters, as was certainly the case with the Phyllini. This slide shows the progression of phyline classification over 4 decades. By 2010, it had become clear that there were at least 5 independent evolutions of ant mimicry in the Phyllini, rather than just one. Oops, sorry. So here we see the progression of classification in the phylines from 1958, Carvalho's catalog, to 2010 with results from a number of authors. In the Carvalho classification, the dissertation work of Katrina Menard using DNA sequences and morphology showed that within the phyliny, there are not just 3 lineages as proposed by Carvalho, or 6 as was the case in 2010, but rather 9 tribes and another 9 subtribes. And you see the two levels above the cladogram here. Of those, no fewer than 5 lineages possess members with a compelling ant-like habitus, here indicated in the red dots, all of those in the subfamily Phylini. Katrina's work also dismembered the classic tribe Felini, which had no distinctive attributes, distributing its members across several lineages, including a primarily holarchic group to bear the tribal name, with numerous remaining genera from around the world placed in Certicetus for a lack of DNA data. The Pulvillian Parampodia provide important characters in the classification of the Miridae and were extensively used by Knight, Wagner, Carvalho, and others, including me. Most Phylini possess cetiform Parampodia, but as here indicated by the green dots, no fewer than 5 lineages possess fleshy Parampodia, which in the feline tribe Pylophorinae are indistinguishable from those found in the subfamily Orthotylini, and were therefore used as a defining character by Carvalho in placing the tribe Pylophorinae in the subfamily Orthotylini in spite of their distinctive male genitalia. As a final example in this talk, in this case regarding the power of phylogenetics, revisionary and phylogenetic work done by Christiane Weirach and me allowed us to offer a credible argument for the existence of transantarctic distributions in the Phylini. Here we see images of some of the taxa involved, although not including images of taxa from New Zealand. The transantarctic patterns had never been known in the Heteroptera, although they are certainly well documented in the Pellurideidae, and are known in many other groups of insects, including midges maybe most notably. Here we see a graphic depiction of the apparent relationships of the southern landmasses as postulated from our work on Phylini. A similar pattern was documented by Eric Gilbert in 2012 for the tinged subfamily Cantacadarini. So in the foregoing, I've attempted to show that existing collections are often inadequate to answer basic questions about true bug diversity in biology, and that for many taxa, only additional focused collecting can provide a clearer understanding of actual diversity and biotic associations. In a complementary way, classifications must be updated periodically to reflect current information, to be maximally informative, and to serve as tests for taxonomic, biogeographic, and other evolutionary theories. Phylogenetic methods offer the most powerful tools to achieve those goals. None of this work would have been possible without the assistance of others, many of whom are listed on this slide, although I may have forgotten others, and funding from the agencies and organizations listed at the bottom. Thank you very much for your attention.
Questions? No one?
John.
Okay, so you showed your very enviable map of all the places you collected in Australia.
Yeah.
And I noticed there was a shockingly low density of dots, both in north central Darwin area and then in the northeast around Cairns. Is that just because of the lack of roadways? Are you spending more time in fewer areas, or were there other logistical issues to that?
Well, there's some logistic issues involved, but Miridae, we already knew by the time I started working in Australia with Jerry that the Mediterranean climate areas of the world showed tremendous diversity, at least in the subfamilies that I was interested in. and that also he was interested in. He'd done a modest amount of collecting in southern Australia, and so that's where we started collecting in the springtime, which is when Mediterranean climates really do their thing, and Phyline miridae do theirs. And that's kind of where we ended up. Jerry's done a fair amount more work in the northern half of the continent, but there is so much diversity in the south that it was beyond my potential fieldwork lifetime, I guess you would say. So that answers part of it. I think the other part is that the fauna of northern Australia is largely tropical or subtropical, and it takes a whole different approach to attack it in a credible way, and I don't think it would have. There's no evidence yet that it would have made much difference in our understanding of miridae phylogenetics. One thing that some people might be interested in is how much interchange is there between Australia and New Guinea, for example, or how much of the Asian fauna, tropical Asian fauna, do you see in Australia? Yeah, the short answer is not very much, actually. Katrina and I worked on one project of mistletoe feeders, of which there are 10 known in Australia now. None had ever been described. There are some known in Asia, so it's a group that has an Indo-Pacific distribution all the way into Africa. But most groups, excluding that one, and the Leucophoropterina, one of the ant-like groups, which also occurs in Australia. Most of the other groups in Australia seem to be largely restricted to Australia. You don't see much interchange between the northern half of the continent, or any part of the continent, and tropical Southeast Asia, including New Guinea. So focusing on the southern half of the continent seemed like a wise decision.
Another question?
Thanks very much.
So now we're gonna go to the coffee break, but before, please, if you could go to the table in the middle. There's a list for the sampling site tomorrow. Just check your name if it's correct or not, so we can end up organizing the buses and cars and everything, okay?